Hello and welcome to this week's module, Gene Expression at the Molecular Level, number 1, Production of Messenger RNAs and Proteins. Cover this process called transcription. The understanding of it, they ended up calling it the central dogma. Okay, so what that's saying is transcription, it produces a transcript or an RNA copy of a gene. This messenger RNA or mRNA specifies the amino acid sequence of a polypeptide. We then have translation. It's the process of synthesizing the specific polypeptide on the ribosome using the messenger RNA template. So it's kind of as if you're passing a note in class, you decide to transcribe a letter in code to your friend gets that note and they have your own little personal code and they translate it to understand what the message is saying so other people don't really know what the note might say. Okay, and the process of this is going from DNA to RNA to the protein. So eukaryotes also have an intervening step called RNA processing, where pre-messenger RNA is processed into active messenger RNA. Okay, and there's the steps for the process. Bacteria themselves do transcription and translation occur in the cytoplasm, whereas in eukaryotes, transcription occurs in the nucleus and translation occurs in the cytosol. Okay, this is just a diagram of that gene expression that we're going to be going over today and the difference between the prokaryote and eukaryote.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
Okay, so exactly how do genes determine traits? Okay, genes constitute the genetic material, which we learned last week, which is called the blueprint for the organism's characteristics. The protein coding genes store the information to produce a polypeptide. One or several polypeptides acts as a protein to play some role within the cell. Activities of proteins determine the structure and functions of those individual cells. Cellular activity taken together in an organism determines their traits or characteristics. They can be anywhere from blue eyes to black hair to the way that you laugh. They basically determine everything. Going with transcription, RNA first forms base pairs with the DNA. Okay, and much like replication, C goes with G, but A goes with U. Okay, so when we're dealing with RNA, U replaces T. So the primary transcript is the length of messenger RNA that results from the process of transcription. So the DNA is going to unfold and the RNA is going to come in and make a copy, okay, and then release a message. So when transcribing, knowing those rules, we can determine what A with C and G go with. ATA, CCC, and TGA. So the gene itself is an organized unit of base sequences that enables a segment of DNA to be transcribed into RNA and ultimately results in the formation of a functional product. Messenger RNA specifies amino acid sequences of that protein. Other genes code for RNA itself as a product. So we have what's called transfer RNA or tRNA, which translates the messenger RNA into an amino acid. And we also have ribosomal RNA, which is rRNA, which is part of the ribosome. So the organization of sequences in a protein coding gene. We first start off with the promoter. This is a sequence of DNA that controls when and where transcription will begin. We then have a terminator, which specifies the end of transcription. We then have regulatory sequences, sites for the binding of regulatory proteins. Some regulatory proteins enhance the rate of transcription, whereas others inhibit it.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex, able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors called coactivators link the basal factors with a third set of transcription factor proteins called activators. The activator proteins are not shown.
Okay, and this is the general organization of the sequences of the promoter, the transcribed region, and then the terminator, as well as the regulatory sequence. First, we start off with this process of initiation. It is a recognition step. In bacteria, sigma factors cause RNA polymerase to recognize the promoter region. The stage is completed when DNA strands separate near the promoter to form an open complex. So we have initiation, elongation, and then termination. We first start off with the promoter functions as a recognition site for that sigma factor. RNA polymerase is bound to the sigma factor, which causes it to bind to the promoter. Following binding, the DNA is unwound to form an open complex. Elongation. This is where RNA polymerase synthesizes RNA. The DNA strand that is used as the template for RNA synthesis is called the template strand. Opposite DNA strand is the coding strand. It has the same bases as the messenger RNA, except uracil is substituted for thymine. It is synthesized from 5' to 3', and behind the open complex, the DNA rewinds into a double helix. Elongation or synthesis of the RNA transcript. The sigma factor is released. RNA polymerase slides along the DNA in an open complex to synthesize the RNA. RNA polymerase slides along the template strand in the 3' to 5' direction. It synthesizes RNA in the opposite direction, 5' to 3' direction. We have termination. So RNA polymerase reaches the terminator. It causes both the polymerase and the newly made RNA transcript to disassociate from that DNA. So when the RNA polymerase reaches that terminator, the RNA transcript, it disassociates or leaves the DNA. That whole process in one diagram. Okay, with the direction of transcription, as mentioned, the direction of transcription and which DNA strand used varies among genes. In all cases, they synthesize RNA transcript from 5' to 3', and the DNA template strand is read 3' to 5'. Okay, with eukaryotic transcription, the basic features are identical to prokaryotes. However, each step has more proteins. There are 3 forms of RNA polymerase. We have RNA polymerase II, which transcribes the messenger RNA. We have RNA polymerase I and II, which transcribe genes that specify non-coding RNAs such as rRNA and tRNA. And we have RNA polymerase II, which requires 5 general transcription factors to initiate transcription or form a pre-initiation complex. Here we just throw those transcription factors together, showing the promoter and the RNA polymerase II. We then have what's called RNA splicing. Here we involve introns, which are intervening sequences that are not translated. We have exons, which are sequences contained in the mature messenger RNA. And the RNA splicing itself is where the introns are removed and the extrons are connected. Introns are found in many eukaryotic genes. We have what's called a spliceosome, which removes enzymes for splicing. And looking at this diagram, this is how the introns are removed and the exons are connected to form that strand. There are RNA modifications in eukaryotes. Bacterial messenger RNAs can be translated immediately. Eukaryotic messenger RNAs are made in a long pre-messenger RNA form that requires processing into mature messenger RNA. This is where the introns transcribe but are not translated, the exons do the coding sequence found in mature messenger RNA, and the splicing removes those introns. Other modifications are the addition of tails and caps. After RNA modifications are complete, the messenger RNA leaves the nucleus and enters the cytosol, where translation can occur. Here are those modifications shown that we mentioned.
Most eukaryotic genes are composed of numerous short coding sequences called exons, interspersed between long stretches of non-coding sequences called introns. The initial messenger RNA molecule copied from a gene includes introns as well as exons. Before this RNA molecule can be used to make a protein, the introns must be removed by a process called RNA splicing. A structure called a spliceosome plays a key role in RNA splicing. The spliceosome is composed of several small molecules called snRNPs, or SNRPs. SNRPs are made up of protein and small RNA molecules. Formation of the spliceosome begins when one kind of SNRP binds to the 5' end of an intron, and a different kind binds to the 3' end of an intron. Additional SNRPs then interact with the complex, bringing the two ends of the intron together and causing the intron to form a loop. This also gathers all of the SNRPs together as the completed spliceosome. The intron is then cut out and the exons are joined or spliced together. The spliceosome then falls apart as the SNRPs are released.
Eukaryotic genes consist of coding sequences of nucleotides called exons and non-coding sequences called introns. The product of transcription, called the primary transcript, is formed with a 5' cap and a 3' poly A tail. The intron sequences are then removed by the process of RNA splicing. The intron-exon junctions are recognized by small nuclear ribonucleoproteins called snRNPs. These SNRPs then cluster together, forming a larger complex called a spliceosome, which carries out the removal of the intron. Splicing involves cleavage of the 5' end of the intron, forming a looped structure called a lariat, joining the two exons together and releasing the intron. One advantage of the splicing process is that a single primary transcript can be spliced into different messenger RNAs by the inclusion of different sets of exons. This process, called alternative splicing, explains how the 30,000 genes in the human genome are able to encode the 120,000 different translated messenger RNAs reported to exist in human cells. On a larger scale, it has been proposed that an advantage of gene splicing may be that during the course of evolutionary history, various combinations of exons may have been created by exon loss, duplication, and reshuffling. Changes in the sequence of the primary transcript could be due to mutations in the DNA of the gene
or to mutations in an intron excision mechanism.
RNA modification with this capping and tailing. Okay, capping, we have 7 methylguanose attached to a 5' end of the pre-messenger RNA. It's needed for messenger RNA to exit the nucleus and bind to a ribosome, and it helps prevent degradation. A poly A tail is 100 to 200 adenine nucleotides added to the 3' end. It increases stability and lifespan in cytosol. It is not encoded in the gene sequence, and it aids in export of the messenger RNA from the nucleus. Diagram explaining the difference between the caps and the tails.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
So transcription is done. What now? Now we have mature messenger RNA transcribed from the cell's DNA. It is leaving the nucleus through a nuclear pore. Once in the cytoplasm, it finds a ribosome so that translation can begin. We know how messenger RNA is made, but how do we read that code?
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
So we move on to translation. Translation and the genetic code. The genetic code, it specifies the relationship between bases in messenger RNA and amino acids in a polypeptide. Read in groups of 3 nucleotide bases, or what is called codons. Most codons specify a particular amino acid, and then you also have a start and a stop codon. You have a degenerate code, which is more than one codon can specify for the same amino acid, and we'll show you what that is here in a moment. Looking at this chart, okay, we can see that we have a number of positions and everything. So what we're looking at is where the UUA, that is going to give us this amino acid. A UCA is going to give us this amino acid. So you can see if we see a pair of UCA or UCG, that's going to give us the same amino acid either way. Machinery of translation. Okay, it requires many components such as messenger RNA, tRNA, ribosomes, and translation factors. Most cells use a substantial amount of energy on translation. Okay. And so here are the components of the machinery that we just mentioned. Okay, so we have the component and their function or definition of what they actually do. In bacterial messenger RNA. Bacterial mRNA has a 5' ribosomal binding site. The start codon is usually AUG. The typical polypeptide is a few hundred amino acids in length. And stop codons, aka the termination or nonsense codons, are either UAA, UAG, or UGA. What we have what is called reading the frame. So the start codon defines reading the frame, read as a group of 3 bases called a triplet. Okay, so as we're looking here, okay, we look for the start codon, and then we start reading the amino acids in triplets. Okay, so we have the start codon here, the AUG, and then we read CAG in triplets all the way down. That gives us the amino acids that is being made. The addition of U shifts the reading frame and changes the codons and amino acid specificity. Okay, so here when we have changed U, we get all different amino acids because of it. Now tRNA. The different tRNA molecules encoded by different genes. tRNA ser carries serine. Common features are a cloverleaf structure, an anticodon, an acceptor stem for amino acid binding. Okay, and we can see those over here. Here we have the anticodon. Okay, we have the anticodon down here, and so forth. Oops. Codons and anticodons. In messenger RNA, a codon is a set of 3 RNA nucleotides. T of DNA is then substituted for U with RNA. tRNA, we have an anticodon, which is 3 RNA nucleotide parts of the tRNA molecule, which allows binding of the tRNA to the mRNA code. And we'll be showing you a video of what that looks like here in a few. We have an anticodon-codon recognition. So question, a tRNA anticodon has the sequence 3' CAG to 5'. What amino acid does this tRNA carry? The anticodon is 3' CAG 5', so it is complementary to a codon with the sequence 5 GUC 3'. According to the genetic code, which is the chart that you saw earlier, this codon specifies for the amino acid valine. Therefore, this tRNA carries valine.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Aminoacyl tRNA synthetases catalyze the formation of charged transfer RNA. To begin the process, a specific aminoacyl tRNA synthetase binds a particular amino acid and also a molecule of ATP at its active site. A covalent bond is formed between AMP and the carboxyl group of the amino acid, and pyrophosphate is released from the enzyme. A specific tRNA that has an anticodon that corresponds to the amino acid then binds to the synthetase. The RNA sequence in the anticodon region, as well as other parts of the tRNA molecule, such as the acceptor stem, are important for recognition between the tRNA and the aminoacyl tRNA synthetase. The bond is broken between the amino acid and AMP the AMP is released. At the same time, a covalent bond is formed between the amino acid and the 3' end of the tRNA, which is now called a charged tRNA molecule. The charged tRNA is then released from the enzyme and is now ready to be used in the process of translation on a ribosome.
Okay, so now we have direction of polypeptide synthesis. So the direction of the polypeptide synthesis parallels the 5' to 3' orientation of messenger RNA. First, the amino acid at the amino end or the N-terminus of the polypeptide. The peptide bond then connects the amino acids. And lastly, the amino acid located at the carboxyl end or C-terminus. Okay, so here we see the sequence of the gene, okay, and we can see the template strands, okay, the coding strand, the template strand, okay, and we can see the messenger RNA, and then the polypeptide being made with the T messenger RNA. It is a good idea to study this slide and make sure you know how it goes from the coding strand to the template to the messenger RNA to the polypeptide. Next, we have ribosomes. A ribosome is the site where translation takes place. Prokaryotes only have one kind of ribosome. Eukaryotes have distinct ribosomes and different cellular compartments. Here we focus on the systolic ribosomes. Eukaryotic ribosomes composed of large 50S and small 30S subunits. Okay, and so here we can see the difference between bacterial and eukaryotic, the small subunit and a large subunit, and then an assembled unit itself. Ribosome structure. With X-ray diffraction used to understand the ribosome structure, messenger RNA lies on the surface of the 30S subunit. Discrete sites for the tRNA binding and polypeptide synthesis are called the P site, the A site, and the E site, as explained in the last video. We then have stages of translation. We first have initiation, which is messenger RNA, first tRNA, and ribosomal subunits assemble. We then have elongation, with the synthesis from the start codon to the stop codon. And then we have termination, which the complex disassembles at the stop codon, releasing the completed polypeptide. Going through that stage by stage, we first have some messenger RNA, seen here, the tRNA, seen here, and the ribosomal subunits, which is the whole complex, which forms the complex. This requires help of ribosomal initiation factors. It also requires input of energy through GTP hydrolysis.
Translation initiation is a process in which mRNA, initiator tRNA, and small and large ribosomal subunits associate with each other to form a complex. Initiation of translation in bacteria begins when the 30S or small ribosomal subunit binds to the mRNA near its 5' end. This process is facilitated by a Shine-Dalgarno sequence in the mRNA, which is complementary to a component of the small ribosomal subunit called 16S ribosomal RNA. Though a shorter sequence is shown here, the Shine-Delgarno sequence is actually 9 nucleotides long. Initiation factor 3 also facilitates the binding of the mRNA to the small ribosomal subunit. Once the 30S subunit IF3 mRNA complex has formed, initiation factor 2 binds to the complex and promotes the binding of the initiator transfer RNA for N-formyl methionine to the complex. IF2 and IF3 hydrolyze GTP as an energy source to promote the association of mRNA, tRNA, and the ribosomal subunits. Translation initiation is completed when the large ribosomal subunit binds and IF2 and IF3 are released. Later, when translation is completed, IF1 is needed for the dissociation of the complex.
Lastly, we have elongation. Okay, so the ribosome travels in the 5' to 3' direction, as seen here. Then synthesis of the polypeptide happens. Elongation factors hydrolyze GTP to provide energy to bind tRNA to the A site. The polypeptidyl tRNA is in the P site, and the aminosyl tRNA is in the A site.
Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP. Once the tRNA binds in the A site of the ribosome, the polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site. Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon. This process is promoted by elongation factor G and requires another GTP. This places the empty tRNA molecule in the E site of the ribosome and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed. Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time.
And lastly, we have termination. The ribosome reaches a stop codon. All of the components disassemble. We have the release of a completed polypeptide. So when a stop codon is found in the A site, translation ends. 3 stop codons, UAA, UAG, and UGA, and these can be recognized by release factors.
Termination begins when a stop codon appears in the A site. Since there is no tRNA corresponding to the stop codon, a release factor binds in the A site. The binding of the release factor causes the polypeptide chain to be cleaved from the tRNA. The polypeptide is released, and then the tRNA is released. In the last step, the 2 ribosomal subunits and the mRNA dissociate from each other. This completes the termination process.
Here we have the stages of translation all in a diagram on their own. Okay, we can see stage 1, 2, and 3. So this is another good diagram to study.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
The difference, we have transcription versus translation review. Okay, so with transcription and translation, transcription is a process by which genetic information encoded in DNA is copied into messenger RNA. This occurs in the nucleus, and DNA goes to messenger RNA. With translation, it's a process by which information encoded in messenger RNA is used to assemble a protein at a ribosome. This occurs on a ribosome, and the messenger RNA goes to a protein.
Let's talk about protein synthesis. Protein synthesis is the process by which your cells make new protein. Let's take the protein insulin, for example. You may recall that insulin is a protein hormone. Insulin is made by the cells of the pancreas, and it acts to lower blood sugar. What happens if you need to make more of this protein hormone? Here's a brief overview of what happens. First of all, the cells of the pancreas that make insulin have DNA inside of them, just like most all cell types do. The DNA in the nucleus contains the instructions for how to make proteins like the protein insulin. Step 1 is to get access to the instructions for making insulin. To do this, the cell has to open up a section of the DNA so the directions can be copied. Step 2 is to copy the section of DNA with the directions for making insulin. The copy of the DNA is called mRNA, messenger RNA. I think of this process as similar to making a photocopy of a page in a book that you can't take out of the library. The DNA has to remain in the nucleus, but the copy, the mRNA, leaves the nucleus and can then be used to make the protein insulin. In step 3, the mRNA finds a ribosome. The ribosome will be able to read the directions and begin adding on the correct amino acids in the order specified. One by one, the amino acids get linked together, like beads being added to a string. Once the ribosome comes to the end of the mRNA directions, the protein has been made. The insulin is ready to be folded into its correct shape and then sent out into the bloodstream to do its job.
Okay, and that is the end of this module's lecture. Don't forget, I started adding those readings to the lecture section. They are not required, okay? They are not worth points. They are just smaller, helpful readings that take this process in chunks, okay? So you can better understand the process in a more simpler manner and then add on the more advanced concepts that we might be going over in lecture. Once again, if you have any questions, don't feel free to email me and have a great day.